Welcome everyone. I'm Dan Akschuti. I'm the biotech analyst at Pareto, one of the life science analysts of Pareto. And it's my pleasure today to present BioInvent, to have Martin Welschof, the CEO of BioInvent, with us. Currently, BioInvent is our top biotech pick, and why is that? It has been dragged down by multiple peers listed in Sweden that had a tough time. They had bad results, and with that, BioInvent stock has been trading down to now a discount that we believe will be very short-lived. So it's currently trading at 40 SEK with a market capitalization of 2.3 billion, while the company has around 1.4 billion in liquid funds. And that becomes even more interesting when we look a bit what is happening in the next months. We expect two bigger triggers in December with already very promising signs towards these triggers. So we believe that they will be most likely positive. And then the next year also holds two additional bigger triggers within the first half. And by thereafter, the pipeline of the company will increase in size a lot. Currently 4 programs, 3 drugs in 4 programs. Next year we'll have 4 programs, 4 drugs in at least 5 programs. And that is a big difference to many peers that are developing only 1 drug. And with that, I would like to hand it over to the CEO of BioInvent, Martin Welschof.
Thank you, Dan. I'm happy to be here and present BioInvent. So this is our forward-looking statement, and I would like to start with a couple of highlights of BioInvent. So BioInvent is a Swedish biotech company, and we are focusing on novel, first-in-class immunomodulatory antibodies, focusing on cancer treatment. And we're listed in Stockholm, and we're currently close to 100 employees. Just a little summary snapshot in addition. So as I already mentioned, focused on innovative immuno-oncology therapies. We are pushing ahead with a portfolio approach. Currently, we have 4 clinical programs running. By the end of the year, we'll start a 5th program. And we're really an integrated company, at least for European standards. So we have a research engine that also would allow for function screening, and we also have our own manufacturing in-house. The platform that we're using has been validated through a number of deals, most recently with Pfizer, but we also have antibodies with Daiichi Sankyo, Bayer, and Mitsubishi Tanabe in clinical development. We're very proud of our strong international shareholders. So our largest shareholder is Redmile. Then we also have blue chip European investors such as Van Herk Investments, then also from the US, Omega, HBM from Switzerland, then a couple of high-quality Nordics such as AP4, Swedbank Robur, Handelsbanken, and then last but not least, Merieux and Invus. We have a very solid cash position, so our current runway will carry us until the end of 2024, maybe into early 2025. So on this slide, you see the core of the company. So this is the platform that we have used to generate therapeutic development candidates for our partners as well as ourselves. And I will not go through this in detail, but just focus on the key features. So first of all, you can see at the top, we are able to use our platform to screen directly on human tumor tissue. And once we have identified our high-quality antibodies from our encoder antibody library, specifically binding to those cells of interest, we check those antibodies on the left-hand side in a number of animal models and focus only on those antibodies that have shown strong therapeutic effects. And for those antibodies, we also identify the target structures. And as I said, so this platform has been validated in a number of collaborations with large pharma partners. And this is the platform that we use or have used to build our own portfolio. And this I will introduce to you now. So starting with a summary of the portfolio. So that was the portfolio in August 2018. And during the last 3 years, roughly, we built it up to something like this. And you will see that we are focusing in 2 areas. So we are focusing on the innate immune system, targeting a structure which is called Fc gamma R2b. And there we have 2 programs. 3 clinical development programs, 2 candidates. One is BI-1206, which is currently in 2 clinical programs, one for non-Hodgkin lymphoma, the other one for solid tumors. And then we have a second Fc gamma R2b antibody, which is called BI-1607, which should initiate clinical development by the end of this year. You can see that we have BI-1206 already some partnering ongoing. So we have partnered with CASI Pharmaceuticals for China, Taiwan, Hong Kong, and Macau. And then we have a clinical supply and collaboration agreement with Merck around BI-1206 in solid tumors. Then the second category is targeting tumor regulatory T cells in the tumor microenvironment. And there we're focusing on 2 target structures. One is TNF receptor 2, and the other one is CTLA-4. And our lead antibody against TNF receptor 2 is also in clinical development, has been initiated by the beginning of this year. And there we also have a clinical supply and collaboration agreement with Merck. And then we have BT-001. This is a combination of our proprietary anti-CTLA-4 antibody with the oncolytic virus platform from Transgene, and that development also has been initiated by the beginning of this year. And then we have a third program, which is called B1910, which is our second anti-TNF receptor 2 program, which is still in preclinical development. So you could see that we have a very, very rich pipeline with a number of very interesting first-in-class clinical development programs. And I will go through this now a little bit more in detail to give you the latest update. So basically, it's a pipeline, a strong pipeline with multiple value drivers. Starting with BI-1206. So on the right-hand side, you see the areas that we're focusing in. So these are 3 subsegments in non-Hodgkin lymphoma, marginal zone lymphoma, mantle cell lymphoma, and follicular lymphoma. And we have a very compelling rationale. In anti-CD20 refractory B-cell lymphomas, we offer a chemo-free regimen since we have a favorable safety profile. And this is basically a first-in-class hematological development program with no direct competitors, with a very high unmet medical need. So coming to the data that we already have generated in that program, and we had a press release last week. So far, we have treated 16 patients with BI-1206 in combination with rituximab. And out of those 16 patients, we could evaluate 12 patients. And out of those 12 patients, we had 3 complete responses, 2 partial responses, and 1 stable disease, which is a very good overall response rate. But more importantly, we have also very high quality of responses. So the complete responses are still enduring. For in one case after 1 year and in another case after 2 years, which I think is very exciting. And that's exactly what you want to see. You don't want to see only a nice overall response rate, but also these quality, long-lasting responses. So this is the study design, and we are in the middle. So basically at the end of the dose escalation, which is part A, and we'll soon go into part B, which is dose expansion. And then on the next slide, you will see the 3 areas that we're really focusing on. So we're selecting the dose for the second part, for part B. Then we're preparing the end of phase 1 meeting with the FDA, where we'll discuss the dose for the second part, and then the study design for the phase 2 part, which potentially could be a pivotal study. So it's quite interesting. And then we work very closely with our Chinese collaborator, CASI Pharmaceuticals, in order to get the China IND by the end of this year. So at the bottom of this slide, you will see that we have also orphan drug designation for mantle cell lymphoma, and we are working on fast track designation and breakthrough therapy designation. So those will be the additional things that we will discuss with the regulatory authorities. The second program for BI-1206 is in solid tumors. So in the non-Hodgkin lymphoma, the previous one, we're focusing on patients who have relapsed anti-CD20 based therapies. In this case, in a solid tumor study, we focus on patients with solid tumors who have relapsed or are refractory to anti-PD-1 or anti-PD-L1 treatment. And here you have a similar design. You have dose escalation and dose expansion, and we are in the middle of dose escalation. So we're actively recruiting patients, and as you can see here, so at the end of this year, in kind of middle of December, we have planned a KOL event during which we'll update on this study, BI-1206 in solid cancer, but also BI-1206 in non-Hodgkin lymphoma. So just before I go to the other programs, here you see the potential application of BI-1206. We're currently in indolent non-Hodgkin lymphoma. From there, we could move into other non-Hodgkin lymphomas, and in the solid cancer study, as you can see here, we're focusing on metastatic melanoma and non-small cell lung cancer. And if you see positive data, we can also branch out into other solid tumors. In addition to the potential in cancer, we also have potential application in autoimmune diseases, which we are currently exploring with academic partners. Then moving to the second part, so those would be programs targeting T regulatory cells in the tumor microenvironment. And the antibodies here are BI-1808 and BT-001. BI-1808 is our TNF receptor 2 program, which is in the clinic. And BT-001 is the anti-CTLA-4 in combination with the oncolytic virus platform of Transgene. And I will go through this a little bit more in detail now. So this is BI-1808 that has very, very strong preclinical data as single agent and in combination with Pembrolissum, which is anti-PD-1. You can see that on the left-hand side in animal experiments that BI-1808 was actually even better than anti-PD-1. And then on the right-hand side, you see the combination with anti-PD-1 in very difficult to treat animal models. Those are animal models that have tumors that don't have any immune cells, and normally they don't respond to anything but to the combination out of BI-1808 and anti-PD-1. We got very good responses. So based on this excellent preclinical data, we went into the clinic, and the study that we started at the beginning of this year looks like this. So here we're exploring both single agent as well as combination. The single agent arm has been started. We're in the middle of dose escalation, and we're planning to give a study update by mid next year. So currently we're treating all comers, and at the end then we will expand into 3 areas: non-small cell lung cancer, ovarian cancer, which is very interesting because there's no immuno-oncology treatment available currently, and then also a rare T-cell lymphoma, which is called CTCL, and which would allow us to go quickly to market because this is a relatively a small patient group and would allow for conditional approval. Then in the combination with Pembrolissum that we do in collaboration with Merck, we're focusing on non-small cell lung cancer and ovarian cancer. So a very interesting study. And as I said, we will do an update by mid next year. So on this slide, you see just the summary. So as I mentioned, this is our second clinical trial collaboration and supply agreement with Merck. We will test 1808 not only as a single agent, but also in combination with Pembrolissum or Keytruda. And I think that will be very interesting. And the FDA has also approved an IND in April 2021. Then moving to BT-001, this is our collaboration, 50-50 collaboration with Transgene. There we're combining our proprietary anti-CTLA-4 antibody, which is more potent than compared with ipilimumab, at least preclinically. We cloned the genes for this antibody in the oncolytic virus, which is vaccinia-based of Transgene. And we do intratumoral infection. And what we achieve is really that by infecting the tumor cells, the solid tumor cells, with the recombinant BT-001, we not only get lysis to the oncolytic virus, but also then at the same time produce anti-CTLA-4 at very high concentrations only in the solid tumor environment. That means we have a very low systemic exposure, and that we also have shown preclinically. And this study also has been started at the beginning of this year. We're currently testing single agents. So we see on the left-hand side BT-001 intratumoral administration at ascending doses. And then we're still in the part A. In part B, then we will do the intratumoral administration combination with Pembrolissum, and then the phase 2A will be several cohorts, different tumor types. And we're planning to have the first phase 1 data during the first half of next year. So on this slide, you will see what I mentioned already at the beginning, though those are our pharma partners. You see Pfizer, Mitsubishi, Takeda, Daiichi, Bayer, etc. And they have antibodies from our platform at various stages in preclinical or clinical development. And those are typical upfront milestone-based and royalty-based collaborations. So they all provided a certain upfront in order to get access to the antibodies. And as those antibodies go through the clinical development in those companies, that will trigger milestones. And in addition, there will be commercial milestones and royalties at the end. So quite interesting, and in any case, a very strong validation of our capabilities. Then at the end, what to look for the remainder of 2021. So in this at December 11 to 14 this year, we'll have BI-1206 in combination with rituximab in non-Hodgkin lymphoma. There will be a phase 1 update at the ASH annual meeting. So that is coming soon. Then in mid-December, we'll update around BI-1206 in combination with Pembrolissum in solid tumors. That will be early phase 1 data. And then we're also planning a KOL event, which will cover both the non-Hodgkin lymphoma as well as solid cancer, which is mid-December. Then we're looking forward to get the approval for the China IND, hopefully during this quarter. And then also working very hard for the CTA submission of BI-1607, which is our second anti-FcγRIIB program. This should also happen by the end of this year. Thank you very much for your attention.
Thank you very much, Martin, for this exciting presentation that you just gave. And just a few questions. So one thing that strikes out to us from BioInvent is actually the shareholder list. You have a unique shareholder list among your peers, where just this year you managed to attract the Red Mile Group, becoming your biggest shareholder at the last directed share issue. They invested $60 million US, around $60 million US. And then you have other specialist funds like HPM in Switzerland, Van Herk, Omega funds, which together hold around 30% of the company. Can you maybe share a few aspects on why they were so interested to invest in BioInvent?
Yeah, so thank you, Dan. Maybe before I do that, just very quickly also to recap, because this was actually a transformational event for the company. So when I joined the company in August 2018, we had around 40% institutions and 60% smaller investors and retail investors, which of course were very loyal with the company and supporting the company for many, many years. But I think we restructured this a little bit through the two most recent financings. So one summer last year and the other one you mentioned Q1 this year. And now we have 70% institutions. You mentioned some of the names. And I think that is quite important, because if you want to really build a company, which in life science, biotech, medical research has to be long-term, you need investors that really have the long-term horizon. And all those investors that you have mentioned, they have that. And I think they have been attracted to the company for three main reasons, I would say. First of all, I think the company, as I already mentioned in my presentation, is very integrated. So we have from the discovery, and that would be targets and antibodies. Then we do our own in-house manufacturing, and then also clinical development. So we can really move very quickly from the stage when we have discovered a new mode of action into clinical developments, since we can generate our own cell lines and produce our own materials. So that's one thing. Second thing is, I think that this combined with very, very good science. It's when I joined the company, Björn Frendeus and his team, they had already built a very interesting preclinical portfolio, all first in class and really top-notch international science. I think that is very important. And then, of course, the team that was already there, because BioInvent is around for many, many years, and you could see that in a negative sense, but you can see it also positive because it means it's one of those teams who have a lot of experience, many, many years in antibodies. They're one of the antibody experts regarding the discovery, regarding the production, and regarding the clinical development. And I think those three components together made investors such as Redmile very, very interested. And that was interesting when we started the discussions with Redmile, because what I was used to, especially in Europe, and I think also in the Nordics, that you just have a horizon for a year, let's say, maybe 2 years, if it really is good. And they came to us and said, well, look, make a plan for the next couple of years. We want to really see what you can do with those ingredients that I just described to you. And that's what we did, came up with ambitious plans, and then they were funding it, supporting it together in this consortium, as you already mentioned. And I think this is really exciting times. And as I said, this is transformational for BioInvent, clearly.
Okay, thank you. That is helping a lot. And could you also, so back then, the first or the second data set, actually, but the first bigger data set for BI-1206, your lead program, came out. And now you had just last week some additional data, very promising data with strong responses. Could you elaborate maybe here what it means to have a patient that you have enrolled in your trial to have a complete response?
Mm-hmm. So in this trial that you're referring to, this is the blood cancer trial, non-Hodgkin lymphoma. And there we're currently focusing on patients who have no other treatment options anymore. So the standard of care, or the gold standard that you normally would use, is an antibody which is called rituximab. And the problem with rituximab, so it has a very good efficacy, but at some time point, every patient, and I mean every patient, will become resistant. So they will not react anymore to this therapy. And then there are other options, but they are very toxic and also would not lead to long-lasting responses. So those patients that we have treated and where we saw good responses are patients who have no treatment options anymore. They were on chemotherapy and targeted therapies, which gave them some efficacy for a couple of months, but also a lot of side effects. So diarrhea, vomiting, and that I mean on a constant basis. And when we give them the combination, BI-1206, which is the BioInvent antibody, together with rituximab, they have good quality of life. And we have seen now 3 complete responses among a couple of partial responses and stable disease. And those complete responses are long enduring complete responses, because normally in those very heavily pre-treated patient population, what you sometimes see is that they respond again, but then maybe just for a couple of months. In our case, we see responses for more than a year. In one case, for more than 2 years, which is basically, you could almost say it's healing. Yeah, so those patients are out of the danger zone, and they don't have these side effects that I already described. So it's quality of life, and then really basically complete responding, which means they don't need to see any other drugs for a number of years at least, which I think is a big success. Absolutely.
So you basically managed to get at least currently 3 patients cancer-free or free of detectable cancer for a longer time, and they're not on drug anymore at all, so they're just living cancer-free now?
Correct. That's correct. Absolutely. And that comes on top of the relatively good overall response. So we have an overall response of around 50%. Of course, I have to say this is still early days, so we have now seen 15 patients. But we're continuing to enroll, and we will also give further updates during the end of the year. But I think the most important thing is really that we see those long-lasting durations. So that means the quality of response. That is really something what we feel is exciting, but not only us, but also the treating physicians and the KOLs that we talk to.
Yeah, absolutely. I think it's one of the biggest challenges of any new cancer drug, including the ones recently approved that are very targeted, like Amgen's KRAS G12C drug, that the responses in many cancers are not enduring. That many patients do respond, but after a short time the cancer comes back ever more aggressive. So that's very promising to hear. To zoom out a bit, how do you see the? We see more and more drugs coming, and of course they work only for a few patients usually. So can you elaborate a bit why this is? Why is cancer such a complex disease to tackle, and how you see the field evolving over the next years with different combinations, different targeted therapies?
Mm-hmm. Yeah, cancer is very heterogeneous. So really depends which cell type, and then also even if they would start from the same cell origin, there could be different forms of cancer. So it's very heterogeneous, and you can in many ways compare it also a little bit to AIDS. The AIDS virus you cannot just beat with one drug, so you need a cocktail. Because what happens, and cancer is in this way no different than HIV. It's once you hit it with one therapy, it escapes, so it develops resistance because a cancer cell wants to divide on a constant manner. That's the program of a cancer cell. So, which means when the immune-oncology assets were developed, anti-PD-1, anti-CTLA-4, anti-PD-L1, so pembrolizumab, ipilimumab. I think there was a big breakthrough for a lot of patients, especially also in solid cancer. But when you look now what has happened, so I think to those type of drugs, even though very good drugs, you have only 10-15% of the patients are responding. So there's still a huge population that does not respond, so which means there's a lot of space for companies such as BioInvent that come up with new immunomodulatory drugs, because that's also what we're focusing on. In different areas. So basically, if you look at the immune system, there are two systems, the so-called innate immune system and then the so-called adaptive immune system. We're working on both. So our 1206, 1607 is working on the innate arm, whereas our 1808, BT001 is working on the adaptive arm. And I think it's important to tackle both. And of course, it's always nice when you see single agent activity. For BI-1206, we have characterized that antibody quite heavily preclinically. For instance, non-Hodgkin lymphoma, there we could see two ways of mechanism, some single agent activity, but mostly tackling the resistance mechanism against rituximab treatment. With BI-1808, it's the same thing. We see single agent activity, but we also see very good synergies with pembrolizumab. And I think that's the future in any way. So even if you see single agent activity, at the end, it will be combination therapy. So I think I would think from my perspective, it's even more important to see good synergies rather than single agent activity, because at the end, you have to combine it. And if you can't combine it because it's in combination too toxic, or you have other problems, then probably your drug will not have a very, very big future. So cut a long story short, so I think it's encouraging and giving you comfort once you see single agent activity, but that probably will not be the final positioning, because at the end you have to combine it. Yeah.
Well, that makes sense. And you mentioned Keytruda, pembrolizumab, having a big success and has carved its way out into different treatment settings, and yet you say that it's only helping around 10% of people. At the same time, it has already gotten to over $14 billion in sales last year. Optivo, I think, around $7 billion. So together, this pathway alone is generating already $21 billion in sales. So there's a lot to still improve there, I guess, if that is only 10%.
Absolutely. And that's also where we are aiming towards with our portfolio that I described in my presentation. So the first, the lead program is in non-Hodgkin lymphoma, which is just a couple of $100 million US market. But all the other portfolol members are tackling solid cancer. And this is, of course, a $1 billion US market. And for instance, 1206 using in combination with Pembrolissum, that we will show the first data by the end of this year. I think if we only could add 10%, 15%, you know, the numbers that you just have mentioned, it's a huge potential. And of course, this, even though, you know, we want to generate value, our main driver is obviously to treat patients with high unmet medical need, and that will also translate at the end into value for the shareholders.
Understood. And since you have now 3 programs actually already active and in solid tumors, plus the other one in liquid tumor, and you plan to introduce a 4th one next year in solid tumor, so you will have 5 clinical programs, so more than most companies in Europe that are not part of big pharma. Can you elaborate a bit on the differences between these programs? Why? I guess it goes again into targeted therapies, but why you're having soon 4 different drugs in the space of solid tumors?
Yeah. So actually, what we have done, because in the past, BioInvent also had a number of programs, but it was very diverse, which means in very different areas, not only oncology, they had activities in AIDS, HIV, but also cardiovascular. So what we did, and that was already started by Björn Frendeus, the CSO, he was really building the science in a more narrow area, which first of all, it was oncology, but not only all oncology, it was immuno-oncology. And they are then basically, as I already mentioned, targeting the innate immune system as well as the adaptive immune system. And the special focus was on the tumor microenvironment. And the diversification that we have, that we're trying to treat, you know, the cells in the tumor microenvironment, whether it's dendritic cells or T regulatory cells, from different angles, because they have different targets, they have different triggers. We look at inhibition, we look at agonizing, so we look at different modalities in order to really tackle them from various angles. And I think also science has shown that the tumor microenvironment is very, very important for the development and outcome of therapy against cancer. And that's our focus. So what we do is we have a narrow focus, the tumor microenvironment, and there we're focusing on mechanism and cells that inhibit the immune system in fighting cancer, and we try to change that. So either eliminate inhibitory mechanisms or cells, or we educate, which means that we turn them from inhibitory cells into activating cells. So basically helping the immune system to fight cancer. And, you know, the initial data that we see gives us comfort and support for this strategy. So I think with this, in some ways, narrow focus, but then widening it in the space, we really occupy this area and have also a lot of synergies between the programs. So we could even think, so currently we're focusing on combination with already established drugs such as Keytruda, and you would have seen that we have 2 clinical supply and collaboration agreements with Merck that provide us with access to Pembrolissum/Keytruda. But you know, finally, once we have more data for our own drugs, we could also think about combining them with each other. But of course, you would do that at a much later stage because currently those are innovative, first in class. You first have to show data, you have to prove that they work. And then in this situation, when you do combinations, you combine with already approved drugs. That makes much more sense from a development strategy.
So very exciting times ahead for BioInvent with at least 4 bigger catalysts over the next, let's say, 8-9 months. Some 2 just in December. Thank you very much for this very interesting chat and presentation.
Thank you very much, Dan. Always a pleasure to talk to you.